Welcome to Bio 22P, Principles of Genetics, exercise number 2B. So for today's video, I will discuss about the M-phase meiosis. M-phase meiosis has cell division or cell cycle. Cell cycle or cell division, it is a series of events that takes place in a cell as it grows and divides. A cell spends most of its time in what is called interphase, and during this time it grows, replicates its chromosomes, and prepare for cell division. So this is the stages of mitosis and meiosis. In mitosis, there are 5 stages, and in meiosis, there is an 8 stages. So in mitosis, the first stage is interphase, second prophase, third metaphase, fourth anaphase, and fifth telophase. While in meiosis, the first stage is prophase 1, second metaphase 1, third anaphase 1, fourth telophase 1, fifth prophase 2, sixth metaphase 2, seventh anaphase 2, and eighth is the telophase. So, in mitosis, interphase, as you can see in this picture, interphase, it is the cell grows and makes a copy of its DNA. During the mitotic M phase, the cell separates DNA into 2 sets, divides its cytoplasm, forming 2 new cells. So sa interphase, cell grows and makes a copy of its DNA. The second one is the prophase. Prophase, it is a chromatin condenses into chromosomes, and the nuclear envelope or membrane breaks down. Membrane breakdowns happen in prophase. Next is the metaphase. As you can see in this picture, metaphase, the nucleus disappears and the chromosomes appear in the cytoplasm of the cell. As metaphase continues, the cell partition into the 2 daughter cells. Next is the anaphase. Anaphase, it is a chromosomes break at centromeres, and sister chromatids move to opposite ends of the cell. Next is the telophase. As you can see in the picture, telophase, it is a nuclear membrane forms around each set of chromosomes to separate the nuclear DNA from the cytoplasm. So let's go to the meiosis stages. First stage of meiosis is the prophase 1. As you can see in the picture, prophase 1. Prophase 1 has the 5 phase or stages. It is the liptotin, zygotin, pakitin, diplotin, diakinesis. The second one is the metaphase 1. In metaphase 1, tetrads line up along the metaphase plate. The chromosomes of the homologous pair attaches to fibers from the opposite poles. Sister chromatids attach to fibers from the same pole. Next is the anaphase. Anaphase 1. The sister centromere remain connected to each other and chiasmata dissolve. Homologous chromosomes move to the opposite poles. So in anaphase 1, the homologous chromosomes move into the opposite poles. So as you can see in this picture, the homologous chromosomes are moving to the opposite poles. Next is the telophase 1. Telophase 1, each complement of the homologous pair reaches the opposite poles. Next is the prophase 2. Prophase 2, it is the chromosomes condense as the centriole move towards the opposite poles. The nuclear envelope breaks down at the end of prophase 2. Next is the metaphase 2. Metaphase 2, each chromosomes align at the metaphase plate. So as you can see, this and this. Oops, sorry. As you can see, these two, this one and the other one, they are lined up. The centromeres of each sister chromatids attach to the spindle fiber from the opposite poles. Next is the anaphase 2. In anaphase 2, sister centromeres detach from each other, allowing sister chromatids to move to opposite poles. And next is telophase 2. In telophase 2, each individual sister chromatids begin to uncoil. The nuclear envelopes reform. So let's go to the product of mitosis and meiosis. The product of mitosis is it produces two identical daughter cells, genetically identical to the original cell, all having two N chromosomes. While the meiosis product, after completing the meiosis M1 and M2, there are four daughter cells produced, each having half of the chromosomes number with respect to the original cells. These cells are called haploid cells. So, let's go to the differences between mitosis and meiosis. In mitosis and meiosis, the difference is in mitosis, it occurs in all organisms except for viruses, while in meiosis, it occurs in animals, plants, and fungi. In mitosis, it creates body cells, one cell division, no recombination, or crossing over, produces two diploid daughter cells. Daughter cells are identical, while in meiosis, creates six cells, recombination, crossing over occurs in prophase. It produces four haploid daughter cells, and the daughter cells are genetically different. So that was their differences, both mitosis and meiosis. And there is their similarities. Their similarities is they create new cells, follow same steps, begin with on parent cell. So next is the life cycle. Life cycle has three types, which is the gametic meiosis, sporic meiosis, and zygotic meiosis. But for today, I will just tackle about the gametic meiosis. Gametic meiosis, this is common in animal and some protista. The term gametic refers to the end result of meiosis, which is gametes. The organism in this life cycle are diploid. This includes the following stages: growth and development of diploid individuals, production of reproductive sea of diploid individuals, formation of gametes, haploid cells, egg or female gametes, and sperm male gametes. Through meiosis, fertilization of gametes, egg plus sperm forming diploid individuals. So this is the example of gametic meiosis diagram. That's all for today. Thank you and God bless.